All this is Dr. Mobeen Sayed. Welcome to one more show. The study today is an in vitro study, but still it almost looked like a Halloween study for me because the important part of the study, or the study in this study, is that the researchers from Sweden found the spike protein to be going into the nucleus, and not only going there, but also impairing, reducing, stalling the DNA repair. This can have drastic side effects. Balance this out during this whole discussion, because when I was reading and going through this, it made me very nervous. So I'm sure that it would make you nervous as well. But please balance that out with this, that we have a lots of infections and we have a lot of vaccines which have spikes as well. And we are not seeing this scale of damage. So what is the actual truth? We do not know yet. It is not an in vivo study, it is an in vitro study, still concerning study. So let's look at this. So I am still working on this new studio, so my apologies for the sound system yet not being 100% and the background and those things. So please forgive me. All right, so here is the study itself. The study is SARS-CoV-2 spike impairs DNA damage repair and inhibits VDJ recombination in vitro. So I'm going to talk about both of those mechanisms and more today. The study is done in Sweden, Stockholm University. Jan is my best friend who is from Sweden. He pronounced it in the most beautiful way. I cannot pronounce it that way. And the UMI University, I hope I'm pronouncing it correctly as well. I have been going back and forth in this study since the morning, trying to make sure that the mechanisms that they're observing make sense, and the data that they have makes sense. So to an extent which I could see, you can see that I have been just going through this the whole day. And my apologies today, Sean is not able to join us. Sean had to actually end up in he had to go to emergency department for some health issues. He's been going through the health issues for some time now, and he will be with us tomorrow, praying for his complete recovery. He's back home, but he's not well enough, and I requested him to take rest, and he would join us tomorrow. So my apologies for that. Okay, so back here. Here's the study. Here's what they found. The study is an in vitro study. So it is not in vivo. It's not observed in animals yet, humans yet. What they found was that spike protein reduces the repair of the DNA. Now, where do we get the DNA repairs? Number one, right now I'm sitting in the studio, there is light shining on me. So my cells, where the light is shining, it is possible that DNA is breaking. When you go to a beach and you sit under the sun, DNA is breaking. When the cells are repairing and when the cells are fighting an infection, for example, SARS-CoV-2, new cells are being formed, the cells gene expressions are opening up and they're functioning, DNA could be breaking during that time as well. New cells are formed, DNA could be breaking and repairing. And then the most important thing, this is the one thing I would request you to hold on to for the whole study's discussion, that is the B cells, when they are making antibodies, when new baby B cells are being made in the bone marrow or the T cells are made in the thymus, their binding region, the region with which they bind with the antigen, those regions are variable. They are different for each B cell. We'll discuss that. That variability is produced by intentionally damaging the DNA and then repairing it. So every time you damage the DNA and repair it, it would form a different shape, and that would give rise to a different variation of the binding region. Now imagine if you're trying to repair, damage and repair and create variations to fight in the immune system, and we could not repair correctly because there is SARS-CoV-2 present, which is stalling and impairing the repair. Imagine the immune system's deficiency. The authors think that maybe that is the reason that some people cannot actually handle SARS-CoV-2 very well, and then we end up dying. Okay, so what is their suggestion? What is the end result of this study? They focused on the vaccines, and they said that it is important to think about maybe not create full spike protein vaccine Instead, in this research, they found out that if you have partial pieces of the spike protein, then this whole thing does not happen. So they said maybe look at spike protein based vaccines, but maybe smaller pieces. I would extrapolate that to maybe not even have the whole spike protein, just the RBD or maybe other parts of the virus. But the researchers' suggestion is in light of this study, maybe full spike protein based vaccines are not the best. So that is their suggestion. That is their suggestion. Um, what kind of issues can be observed? We just went through some of those. People who may be recovering from cancers, maybe recovery from cancer means what? There are cancer cells which are now being told not to survive, or they are learning to repair, or they're becoming better, or we are killing the cancer cells and the remaining cells are becoming better. And so there is cell division happening and there is damage happening. And if that damage cannot be repaired correctly, we have a problem. Similarly, newer cells formation, for example, in the younger ones, new cells are forming all the time, and that means there could be the DNA repair issues which are taken care of. We all have DNA repair happening all the time. Even now, right now, all of us are getting some DNA repairs at various parts. So I want to put this in front of us. How do we prevent this? We prevent it, according to the researchers, by not creating vaccines which are full spike protein. That also means don't get infected, because infection is also full spike protein. And maybe use smaller subunits of the spike protein, for example, RBD, or other parts of the virus. Now, what are their findings? I'm going to go through this. Now we are actually going into the study's details. If you just wanted to understand that what the study says, we're done. Now let's see what are the findings. We're going to go in the details. So for the findings, couple of things that we need to be on the same page with, and that is, we know that when a virus arrives, the SARS-CoV-2 we are talking, that is the context. When I say a virus, that is SARS-CoV-2. We know that when SARS-CoV-2 arrives in our cell, its messenger RNA starts becoming developed into proteins by the ribosome, our cell's ribosome, correct? Then that ribosome makes pieces of the SARS-CoV-2, including spike protein, and then the non-structural proteins, which are called NSPs, and then structural proteins like matrix protein and others. So a bunch of things are made that are all for the virus. Then when all of those proteins are made, they are sent into endoplasmic reticulum, where they are kind of put together. This is like making a car. So you make the doors in one place and the tires in another place and engine in some other place, and then you put them together in one place, and then you give it to the Golgi operators, which would package them up, and that would then put a membrane on top of this structure, and that would become an enveloped virus, and then that virus would get out of the cell. This is the normal behavior of the cell replication. Now, this spike protein, when it is produced, either because of the infection or because of the vaccine, when it is produced, this is the first time they're showing it, that the spike protein ends up inside the nucleus. Not only spike protein ends up inside the nucleus, NSP, non-structural proteins, ends up in the nucleus as well. And not only they just end up there, not only it's just contaminant that has entered this nucleus, they actually go in there and interfere with the machinery of the DNA, with the repair system of the DNA. So that is the thing to keep in mind. So back here, there is spike protein that localizes in the nucleus, goes to nucleus. There are non-structural proteins. These are the enzymes of the virus. NSP, non-structural protein. There are various enzymes of the virus that help virus replicate and make new viruses and so on. So virus has some machinery. This is that machinery. These also, the NSP1, 5, 9, 13, 14, and 16 also end up in the nucleus. My apologies. This is Luffy standing and scratching the door outside. Maybe I should just open up the door for him. So give me one quick second. My apologies. So the door is open. Everyone is now a fair game to come in. So co-location occurs, correct? Now, What are the mechanisms to observe? And the cat is here now. So it is going to make its appearance in front of the camera anytime soon. So here is something to keep in mind. First concept to keep in mind is double stranded break. Double stranded break, we know that our DNA is kind of woven like two strands together, correct? So when both of those strands are broken down, that is called a double strand break. Sometimes it is just the one unit, one strand breaks down, not both. So let's say this is one strand, and let's say this is the other strand. Sometimes it is just one strand that breaks down, and the repair would need to be repairing one strand. And sometimes both strands break down. That is called a double stranded break. So double stranded break can occur. It happens all the time, and especially in the immune system cells when they're making their binding regions, double stranded breaks are intentionally created. Secondly, this is a cell cycles diagram. A cell cycle, a cell that is dividing. It goes through various phases of division. So important thing to note, just to keep in mind here, G1 phase of the cell cycle, if there are DNA breaks that occur during this time, and please remember this, when our cells are dividing, there are strict mechanisms to make sure that the DNA is correctly repaired and correctly copied, and there is no damage. Otherwise, the cell will become a cancer cell. We have an elaborate mechanism to repair DNAs. And so, through this cell cycle, we stop the cell in various cycles. This is like if you're making a building, you stop the building at various milestones, and you say, we'll send inspectors in who are going to look at the building built so far, and they're going to tell you, give you a feedback. And if there is anything that is not correct, correct it before you continue. So at every cell cycle phase, the cell is stopped. Then repair inspectors come in, they inspect the cell's DNA. Then the repair workers come in and they repair it, enzymes here, and then they give a green light that okay, you can go to the next phase of the cycle, and this is how the cell cycle occurs. So if there is a break that occurs during G1 phase, and we can talk separately what are the cell cycle phases, then NH, non-homologous end joining repair mechanism is used, EJ. If the cell is in synthesis, S or G2 phase, growth 2 phase, then HR or homologous repair mechanism is used. So just keep in mind that there are multiple mechanisms for DNA repair because there are multiple kinds of repair damages. And so these two mechanisms are important to keep in mind because these two mechanisms are impaired by the spike protein's presence. Okay, continuing. Now, one more mechanism to keep in mind. So I'm just putting the context together so that then I can show you how the damage occurs. The second mechanism is proliferation. Those of you who have been with me throughout this 20 months now, you know that when the infection occurs, any infection occurs, what happens is that cells, B cells and T cells, they proliferate. Proliferation means increasing in number. Increasing in number definitely means making copies of the DNA, which definitely means there could be damage to the DNA, which definitely means there is going to be need for repair. Plus, the proliferation itself is an important immune system response. This is why some people's lymph nodes would start swelling and paining or aching or tender. Why? Because there are cells that are increasing in number inside of them. This increase in number is called proliferation. So please keep this in mind as well. This is one cell. It gives rise to multiple daughter cells. Then those daughter cells give rise to more daughter cells and so on. And the proliferation occurs. So please keep these. So here, this is a B cell, and it has proliferated in the presence of infection, let's say SARS-CoV-2, and given rise to multiple B cells. Then here is a T cell in the presence of an infection or a stressor or a pathogen. It has increased in number as well. So proliferation has occurred. All right, continuing. There's a lot to be discussed. I'm still talking normal, and we'll talk about abnormal in a second. Now, variable region. This is the important part. Look, our B cells. Imagine if we have 1,000 B cells standing in line, and we ask them to show their hands. All of their hands, every B cell's hand will have a different pattern to grab. Their hands are not like our hands that can be adapted. they are given one fixed structure. So some B cell would be looking like this, another B cell's hand would look like this, somebody's hand would look like this, somebody's hand would look like that. So all of their variable regions are different. So here in this diagram, what am I showing? Look at this part. This is an antibody. An antibody, we have been talking about it from ever, has a heavy chain. These are proteins. So heavy chain is a big, heavy protein. This is another heavy chain. Then it has a light chain. This is the structure. This is another light chain. At the end of the heavy chain and light chain, there is a variable region. So imagine one antibody. Imagine me as an antibody. Every one antibody has two arms and that have two hands, and they both have identical binding sites. So these are the binding site. This part which binds with the antigen, which binds with the offender, which captures a prisoner, what is that? Criminal. That part is called variable region. Formation of this variable region needs DNA to be broken down and repaired, and whenever it is repaired in weird ways, there are weird variable regions that are formed. On this side here, this is a T cell receptor. Now, just like B cell need to bind to an antigen to function with it, T cell also needs to bind with the antigens to function with them. So this is a T cell receptor that also has a similar binding site, and then it has a constant part as well. The structure is slightly different. It has alpha chain and beta chain, not here like heavy chain and the light chain and then further parts of them. It has a alpha chain and a beta chain. Alpha chain in turn has a variable region and a constant region. Beta chain also has a variable region and a constant region. The T cells variable region is made in the similar way by changing and modifying the DNA. So what is the repeated pattern I'm explaining here? DNA repair, break and repair can also be done in immune system cells intentionally for normal function of the immune system cell. Right now, in all of us, there are B cells being formed in our bone marrow, and there are T cells probably getting trained in thymus or some other place in adults. We don't exactly know, but let's say thymus. This is cat, not me. So these B and T cells, when they're forming, and then there is a binding region of theirs, which all of them have, right now, there are DNAs that are being broken and then repaired. And it's a normal thing. So example of the variation. So imagine these are variation or variable parts of various T or B cell receptors. So this part here, this variable region or binding region can bind to an antigen of this shape. This part can bind to an antigen of this shape. This part can bind to an antigen of this shape. And here, if this is a bacteria, bacteria has some patterns on its surface. And look at this T cell receptor. I have removed the T cell, just the receptor is there. So the T cell receptor over here has bound to this antigen. Now to this antigen structure, of course, the other guys will not bind. I hope this becomes clear that every B and T cell needs a different variable binding site. And to create that variation, we need DNA to randomly restructure, which needs DNA break and repair. Then here is the actual DNA break and repair, which can happen for other reasons too. As I said in the beginning, radiation causes it, chemicals cause that, heat or cold can cause that, physical injury can cause this. So there are many reasons that DNA can actually break. DNA, new cell production, cell cycle, cell growth, all of those things can cause it. New sperm formation, new ova formation, although ova are always present in the beginning, so they're not newly formed. Sperms are formed every time. So there are many such cases where new DNAs are made and where DNA repair may be needed. Now here is an example of a double-stranded DNA break, where let's say this was a normal DNA and this part got deleted, and so now both strands of the DNA are broken. This is called a double-stranded break. This kneading is my cat. It's Kyrie actually today. It's not Luffy. If it was Luffy, Luffy will do meow. Okay, so here, imagine this is the DNA, and this DNA has a double-stranded break over here. If this break occurred, as I said earlier, during the G1 phase, then non-homologous end joining repair type will be used. If it is synthesis or G2 or growth 2 phase, then homologous repair mechanism is used. But for both of those mechanisms, there are certain repair enzymes. Imagine there are repair workers in our body, in our nucleus, that would rush to the place of DNA injury, DNA break, and go and fix it. And there are different kinds of repairing enzymes. So, two important enzymes that these research workers looked at, one is called BRCA1, and the other one is 53BP1. And I have references in the description. You can go and see the diagrams. The point of these enzymes is that wherever there is the damage, they would go and attach there. They would create a focus there. They would mark that place. Have you seen the road construction workers? They will go to the road and they'll mark various places where the next set of workers would come and work on. Similarly, these enzymes, BRCA or 53BP1, they create foci. They create the focuses on the DNA's damaged pieces. They inspect the DNA and say, well, it is damaged here. I'm going to put a flag here. Then the repair guys would come in, the enzymes, and they would repair that part. Now imagine if these two enzymes cannot do their function. Imagine if they cannot even be produced. Who's going to repair? Who's going to mark the area for repair? So this is like we have a pothole in a road. Road is damaged. We want someone to come and put a flag to say it needs repair here. And then the repair people would see where the flag is and repair. Imagine the repair flagging enzyme cannot go and put the flag. There's going to be no repair. Okay, so these are the primary normal mechanism. Now let's see what researchers found. What researchers did was the following, and I'm going to go through the same mechanism again and show what was the problem they saw. First, cell proliferation. The researchers found, look at my sneaky way of putting these things down here. Researchers found that the non-structural proteins number 1, 5, 13, and 14, when they go in the nucleus, I mean, this is the first thing, why are they going in the nucleus? But when they go in the nucleus, I mean, just accept the fact that, yeah, they go to the nucleus. Okay. When they go to the nucleus, and how did they do this in the research? They took, this is an in vitro study, so they took cells, they injected in the cell the messenger RNA and DNA pieces to make these structures. They did not infect the cells, they just put these specific proteins and spike protein RNAs in those cells, they transfected them. And then they observed the proteins being formed. Then they tracked those proteins where they are going. Why did they not just infect them with the SARS-CoV-2? Because SARS-CoV-2, if infecting, it is going to destroy the cell. They want to transfect a cell with more benign proteins, and then they want to track the proteins inside the cell to see where are these things going. And then they want to see what is the abnormality of the function of the cell. So they cannot do that by infecting the cell, because the SARS-CoV-2 is just going to destroy the cell. So here they put the plasmids in the cell which had the genes to produce these proteins of the SARS-CoV-2. When those proteins were created, overexpression of those proteins was observed, and this was intentional. It's in vitro. Result was what? Reduced proliferation of the cells occur. So how do we, from in vitro to in vivo, observe that? What this means is, if this is all correct and reproducible in vivo as well, then what that will mean is that when the SARS-CoV-2 is present inside a cell, the SARS-CoV-2's proteins are going to go to the nucleus. And when this cell, let's say it is a B cell or a T cell, and it is trying to divide, the proliferation would not happen or will reduce. Now, there is a lucky thing, and they say it, authors. SARS-CoV-2 is not known to infect B cells and T cells. So that is the lucky part. How did they observe it? They actually transfected them, or not B and T cells. They took cells they made these cells look like B and T cells. So good news is B and T cells actually do not get infected by SARS-CoV-2. Otherwise, we'll have a problem more disastrous than HIV. I mean, imagine a respiratory virus, which is also HIV-like attacker of the immune system cells. Will be done. So it is not that. However, they say that it is known, and they have cited the studies. They say it is known that when SARS-CoV-2 infect a cell, let's say not a B cell or a T cell, some other cell, and it is making its proteins in that cell, it's making more viruses in that cell, parts of the genetic material of SARS-CoV-2 can leak out of this under stress cell. This is called exosome leakage. Exosomes are part of the DNA, RNA material. So some genetic structures, some recipes can leak out of the cell. And these RNA pieces can enter the other cells, which could be B or T cell. If they do enter there, and if they go make spike proteins in them, then we can have a problem with the B and T cell. So it's a far-fetched idea, but this is how they said can be possible. And you could actually throw out this whole study on the basis of this one mechanism problem, that SARS-CoV-2 luckily is not known to infect B and T cell. So this theory over here, this observation that they did in vitro by transfecting the cells, is actually not possible in vivo. But we also didn't think that the spike protein will go in the DNA. So I'm going to keep myself open to understanding more of this part, or this study. Of course, there will be more studies that would try to replicate this, and they would report further. Even maybe the same researchers would do more work. Okay, so back here. As the NSPs, non-structural proteins, are increased, they go to the nucleus, they affect the proliferation parts of the cells. If that happens in the immune system, then the immune system function will reduce because we cannot have enough B cells and T cells. Okay, moving on. First observation from the study, again, in vitro transfected cell studies, that cell division can be impacted. Now, please keep in mind, if the cell division is impacted for B and T cell, and that is not possible in vivo, there are other cells whose division is needed. Maybe that is affected. Growth of the cells, sperms, many other cells are dividing, red blood cells and so on. Stem cells in the bone marrows are dividing, right? So back here, now the second part, the variable region production, which is also very important for immune system function. Now, to produce this variable region, as I have been kind of saying it again and again, that is, what happens is, look at this genetic material. Imagine this is the recipe to make an antibody or to make a T cell receptor. What is important to keep an eye on at this time is that there is a constant part of an antibody or a T cell receptor. We forget about that. It is a variable part as well. That variable part is this part. That variable part is the one that binds to various patterns. Here is how that variable part is produced by breaking and repairing DNA. Here in the beginning is the DNA recipes. These are called V region, D, and G regions. These are genes. This is genetic material. V, D, and J. Some pieces of this genetic material are taken from V, some from D, some from J. This is equal to, if I ask you to cook some food and I give you a book and every page has some recipe on it, and I say, up to you, do not just make one page's recipe. Instead, take 2 instructions from any one page, then take 2 instructions from another recipes page, then take another 5 or 7 instructions, totally up to you, from a different recipes page. Just open the book wherever you like. Then combine those all statements that you read and just cook according to that. Whatever is formed is my luck. This is how it happens. So we take some statements from one recipe, some more statements from another recipe, some more from another. We don't take a whole recipe, just take some statements. Then what happens? How do we take some statements? We delete everything else. So instead of you reading 2 lines from a recipe, what you do is you delete everything else to leave 2 lines in there. So that means you're tearing the book. So here we intentionally are damaging the DNA to leave parts of the DNA. Then those parts are collected together and made into a variable region genetic material. But to put them together, we need repair. This is an intentional DNA damage and repair. This is somatic hypermutation. This is the variable region formation. This is what protects us. This is what protects us from the infections. So it is a good thing. We need it. But this repair needs the repairmen. And we just saw that spike protein could cause the repairmen not to function correctly. That means the variable region generation for B and T cell will not occur very efficiently. That means our capability to respond to infections will not be good. So now immune system cannot have more cells. I'll keep reminding the audience that it is in vitro, and immune system cells are not known to be infected by the SARS-CoV-2. So making this in vivo will have to be a far-fetched idea. If this happened, then the variable regions will not work correctly. And the authors say that why did they think of doing this? That is because they observed people's immune system not to work correctly, and that made them think that maybe there is something in this virus that is impacting their immune system. So even when we do not know that this happens, maybe it is exosome, maybe we'll find out it actually does affect them. Anyways, so this is, I hope this point is clear. We create variable regions by break and repair. And if we cannot create variable regions successfully, then immune system cannot function successfully. Second part, then it is Kyrie, which is just running around here. Third part, third abnormality. These enzymes, now please realize what happens is these enzymes are produced in the cytosol, in the cell's cytoplasm. How? Let's say this is a cell and it needs the DNA repair enzymes. Here is how it works. From inside the DNA, the recipe to make these enzymes will be copied in the form of messenger RNA. That messenger RNA will be sent outside of the nucleus because inside the nucleus there is no mechanism to be able to translate messenger RNA. So even when nucleus needs its own machinery, it still has to request the people outside of the nucleus to say, can you please make me enzymes? So they would say, what do you want? And nucleus would write down, I need following machinery to be made. How does it do that? It sends messenger RNA outside. So please remember, if the messenger RNA went in the nucleus, it doesn't do anything. Even nucleus's own needs are written on a messenger RNA and sent outside because inside it doesn't do anything. Outside, this messenger RNA goes to a ribosome. It would work with the ribosome and the enzymes will be produced. Those enzymes will then go back in the nucleus and go and function there. And what did they find, the researchers? They found that these repair enzymes, when they were being produced outside and they wanted to go in the nucleus, spike protein's presence in the nucleus reduced the production of messenger RNA, which would go and get the machinery made to come in. Wicked. Clever, wicked. That the spike and other proteins are actually blocking the nucleus from forming the machinery to repair itself. And so what they observed was, in this study, that only with the full length spike, not with the pieces of the spike, and they did this in the research. They took pieces of the RNA as well and kind of made pieces of the spike. They created plasmids that could only have enough genetic material to make pieces of the spike and not the full length. Only in the full length spike, the cells, those cells that were transfected with the genetic material to create a full length spike protein, they had marked inhibition of production of BRCA and 53BP1 and their function. That means the repair focus was not present. That means DNA repair was not occurring. This doesn't matter what kind of a cell it is. Any cell that has spike protein in it, if it needs repair, its DNA's repair, then spike protein can reduce the DNA repair. So now the question, this is the end of it. Now the question is, if you said, all right, dangerous and scary, what will happen to the cell? Normally a cell that cannot repair its DNA, usually that cell dies. It would commit suicide. First, it would try to fix it. If it cannot fix its DNA, usually it commits apoptosis and dies. But we know there are cancer cells. Cancer cells are the cells where the DNA, I still have to figure out where the mouse is, where the DNA has escaped the repair, it has become damaged, and it has just started building a weird kind of a cell that is not responding to any controls, and that becomes cancer. So there is even if we dismiss this research to say, well, B and T cells will not be infected in vivo, so we should be okay. There still are two risks. One is exosomes can go into the B and T cell, and the second one is that the dividing cells or repairing cells, any cell, in the presence of SARS-CoV-2 proteins, either with the infection or with the vaccine, can end up in trouble. And so that is the discussion. I hope that it makes sense. Somebody made a comment, this is the last thing, and then I'll stop and we'll come back with the chit chat. Somebody made a comment that I only share studies that are a specific type. And I feel bad that I have been for 20 months sharing all kinds of studies, pro-vaccine studies and vaccine side effect studies. And so I cannot stop people from labeling me, but still, when somebody wants to say I do not do something, they should at least look at my list of lectures, which is I think about 1,000 now, to see have I covered topics. And if they cannot, then it is just a very lazy thing to just put their comment out there. Anyways, with this, let's stop. Please do me a favor. If you can, like, subscribe, and share. That would be awesome. If you don't want to do anything, but you enjoyed this or you had use of this, then at least like this. And if you would like to support this work, then there are 3 links in the description. You can buy me a coffee, or you can be a patron, or you can use PayPal. And Enemy is quite close to me. Tomorrow I have a big day because we have one of the auditors providing their feedback to us. So they're trying to shut down everything. I'm trying to stay afloat. So anyways, all your support is welcome. And thank you very much. I'm going to hang up now and come back for a chit chat.